In this video, we'll be having a look at glandular epithelia. So let's start off with the definition of a gland. So the structure is of epithelial origin, and it makes and secretes products from designated secretory cells. So it arises from epithelial tissue that migrated into deeper connective tissue instead of remaining at the surface. So embryologically, its roots are really tied in with epithelial. It's classified either by the shape or by how products are released. And products are released by two methods. Endocrine, so we have a series of glands called endocrine glands, and a series of glands called exocrine. So let's start talking about endocrine glands. And they secrete products. Many times they're hormones, and they secrete them directly into the bloodstream. Now here's the big point: without the use of a duct. Now, think about it. If you're going to put products into the blood goes everywhere, almost everywhere, so you're going to distribute the products throughout the whole body. So allows products to have a systemic effect on distal cells. And this is often tied in with the concept of cell-to-cell communication, which is one of the core principles. Let's look at exocrine. Exocrine, what they do is they release their products onto the apical surface of the epithelium. So we're talking typically about the external surface of the body, or into the space in a hollow organ, which then leads to the outside of the body eventually, usually. So the products that are secreted from the gland travel through ducts. Okay? And they only have local effects on the cells near the gland. So you can tell there's already a difference between endocrine and exocrine. Now, the glands can vary quite a bit in terms of their complexity, from a single cell to large multicellular glands with a whole number of branching ducts and many secretory units. So we can go from very simple to very complex appearance of these exocrine glands. Now, one of the cells that we can talk about associated with this are goblet cells, and we actually talked about goblet cells very briefly in a previous video. We had a look at them in a certain tissue. This is the most common unicellular exocrine gland. And where do we find it? We find it digestive and respiratory tract. And look what it does. It secretes mucus. It makes sense. The respiratory tract, I want some mucus in there that will trap particles, bacteria, etc. And therefore I can use cilia to move it up and out. And then in the GI tract, it helps to move things along in the GI tract, in certain parts of the GI tract. So goblet cells, they secrete mucus, and they're very distinctive in shape. When you look at them histologically, you know, someone imagined that they kind of look like a goblet. That's how they got their name. And look at, you can see that they're full of mucus. You can see that the material inside the goblet cell is quite different than anything in the neighboring cells. And you can also see that it opens to the outside world in this view. And if we look quite closely, we can see here sort of a typical goblet cell. And there's a nucleus, there's rough ER, there's mitochondria, there's Golgi, and we have cytosol, but most of the inside of the cell is made of the mucus droplets, which are released. And as often when we see goblet cells, we'll see microvilli, and also we'll see tissue with cilia to help move the mucus along. Now, most exocrine glands are multicellular. And here we use a different type of classification. We classify it according to the structure of the duct and the shape of the clusters of the secretory cells. That's going to make a bit more sense shortly. So, in terms of the duct structure, simple glands. Oh, now, simple means simple, right? The ducts don't branch. And compound glands, they have branched ducts. We'll have a look at that. So remember, we can classify them based on the structure of the ducts. So it could be simple or it could be compound. Also, we can describe it based on the shape of the clusters of the secretory cells, the cells that are actually making the product that's being released. So clusters of secretory cells, there's 3 possible configurations. Tubular, that one makes sense. Long, straight, or coiled. Acinar, it's spherical in shape. And then we have one which is tubulo-acinar. Now you notice that sounds familiar. Tubulo, like tubular. Acinar, like acinar. And the reason is, it's a combination of a name, is because it has both tubular and acinar sections. So the 3 types are tubular, acinar, and tubular acinar. And here we can have some, sort of get a visual look at it. So here we're looking at a multicellular exocrine gland. And we can see that here, the duct, there's a duct. So these are ductal epithelial cells. But down here, these are the secretory cells. Right? Now, if the duct is just straight, then I call it simple. That's one of the words I'm going to use to describe that gland. If it's branched, I'm going to call it compound. Now remember, the other thing that I look at, and when I'm come up with a name, is I also look at how the secretory cells are arranged. Right? So tubular means that they're just in sort of basically a tube shape. Acinar, sort of rounded, and tubular or cinar, you can actually see there's this enlarged rounded end. But here, the cells that make up part of the tube are also glandular cells. They're actually making a product. So we can have the tubular, where the cells that are making the product are in part of the tube. We have other ones where at the end you get this sort of enlargement of cinar, and then we have some that where glandular tissues here, but also these are cells that make product. And you can start to look at, as we sort of put the words together. So here's one that I'd call simple tubular, because it's not branched, and the actual cells that are making the product are part of the tube-like structure. Simple acinar, simple once again, because the duct, remember these duct cells are not making a product, they're just there to design to become a tube to carry the product. These are the cells that make the product, and because they're arranged in a circle, I'm going to call them acinar. So this is simple acinar. Then I have compound acinar. Remember, compound means the ducts branch. So definitely this is a compound gland. And because at the ends, I have this rounded collection of cells. Remember, these are the cells that are making the product. These cells, in this case, are just there to put together, make a tube to carry stuff. They're not making the product. And so that's a compound acinar. And then I have a compound tubulo-acinar, because let's really try and zoom in a little bit more on this one. You can see that definitely this part looks very much like the acinar shape that we're talking about, sort of that rounded. But part of the tube structure, not all, but part of that tube structure are still cells that are making the product. So this is compound, right? Because it has all these shapes. And then, because some of the cells that are making up part of the tube structure are producing the product, it's tubulo. And because many of the cells at the end here, are forming this big round area, acinar, so this is an example of a compound acinar. So those are some of the basic names when it comes to glandular epithelium. And remember, we base it upon, is it branched or not branched, the tube? Okay, that's heading out. Also, the secretory cells, are they in a tubular shape, a acinar shape, or a combination, which is tubular acinar? The next thing we could have a look at is the concept of merocrine. Now, these are interesting ones. This is an exocrine gland. So merocrine is used by the majority of exocrine glands, and that would be things like salivary sweat glands and what happens is the products are packaged in secretory vesicles for release by exocytosis into the ducts. And we can see this in miracrine. So inside these cells, I'm making the product, I'm putting it into vesicles, the vesicles being brought to the edge, and then the vesicles release their contents into the ducts here. So I'm going to use exocytosis for that. It's a type of exocytosis. Now here's a different one. These are, once again, the exocrine gland. These are holocrine. Now, sebaceous glands, they use this method to secrete something called sebum. And what happens is here, the secretory cells just keep making product and storing it into their cytosol. And how does the material that they made get out of the cell to be used? Well, they do not use exocytosis. What they do is the cells basically, let me zoom in here, the cells make the product, they keep filling themselves full of product, and then what happens is they shed into here, and then they break down. And so basically they end up releasing the product. So the product's only released when the cell ruptures and the cell dies. So that means if I'm going to be losing these cells on a constant basis, I'm going to have basically stem type cells which will undergo mitosis to replace the ones that are shed. So just a quick review of that. Miracrine, you make the product, you put in granules, the granules move to the edge, or vesicles I should say, move to the edge, and through exocytosis they release the content. That's miracrine. Holocrine, the whole cell fills with material, the cell sheds, and then the cell, I should say, ruptures, releasing the material. Now, there's another one called apocrine, and it's a rare type of secretion. And what happens is portions of the cytoplasm get pinched off and the product's being secreted. And we can see this with lipid droplet secretions in things like the mammillary glands.